Okay, so welcome back to this next video in which we are discussing vasoactive intestinal peptide and pituitary adenylate cyclase-activating protein receptors. Okay, right, so we've now discussed the G protein cycle in full generality. Okay, we now want to get down to specifics. Okay, we have these three receptors, VPAC1, VPAC2, and PAC1, which are these three receptors which are considered VIP and PACAP receptors. Okay, now as we've seen, only VPAC1 and VPAC2 are actually really receptors for both vasoactive intestinal peptide and pituitary adenylate cyclase-activating protein. Okay, but PAC1 does have some affinity for VIP. It's just 100 times smaller than the affinity for PACAP. Okay, and remember also that these two are also receptors for the two related peptides to VIP, which are peptide histidine methionine and peptide histidine valine, PHM and PHV, whereas those aren't ligands for PAC1. Okay, right. So we know that all three of these are secreted in family G protein coupled receptors, and therefore when these ligands bind to those receptors, they're going to cause conformational change that will now allow these receptors to interact with heterotrimeric G proteins. But which heterotrimeric G proteins are they going to interact with? Well, all three basically interact with GS heterotrimeric G proteins, which means that they interact with heterotrimeric G proteins where the alpha subunit is a G alpha S subunit. Okay, so we now want to see what is the downstream signaling pathway of these alpha S subunits once they have been activated and have GTP bound, because as soon as these ligands bind to their receptors, the receptor will then bind these GS heterotrimeric G proteins, catalyze the conversion of the alpha subunit from the off state to the on state. The beta gamma complex will then cleave off. The alpha subunit in the on state will then cleave off. And we're not going to dwell too much on the beta gamma complex signaling. We'll mention that later. But the alpha S subunit with GTP bound is going to go and activate adenylyl cyclase enzymes. Okay, so this is the next big topic that we need to discuss, adenylyl cyclase enzymes. Okay, right, so there are 10 human adenylyl cyclase enzymes. Only 9 of them are going to be important for us to discuss. Okay, so there are 9 what are known as membrane-bound adenylyl cyclases. Okay, and these are the important ones. Okay, as far as our discussion is concerned. There is one final adenylyl cyclase, the 10th adenylyl cyclase, which is known as the... Oh, and by the way, I should have said, adenylyl cyclase enzymes are often abbreviated to ACs for short, A for adenylyl and C for cyclase. Okay, there is one final adenylyl cyclase enzyme known as the SAC, which stands for the soluble adenylyl cyclase. And what this means is basically it's not membrane bound. Instead, it's just within the cytoplasm. Now, this one is a bit of the lone one, basically. It's different to all the other 9. All the other 9, I can draw one picture and that summarizes all of them, whereas this one's different. Luckily, however, this one is only found really in the testes within boys. Okay, so we don't need to worry about this one at all. So we're going to focus on the 9 membrane-bound adenylyl cyclase enzymes. And I think I'm going to go over the page to start the drawing of the picture because I can't really fit it in down here. Okay, but before we do that, let me just tell you that the 9 membrane-bound adenylyl cyclases are called adenylyl cyclase 1 all the way up to adenylyl cyclase 9. So there is adenylyl cyclase 1, adenylyl cyclase 2, adenylyl cyclase 3, adenylyl cyclase 4, all the way up to adenylyl cyclase 9. And then soluble adenylyl cyclase is viewed as adenylyl cyclase 10. Okay, right. So, Let's now draw a picture of all of these membrane-bound adenylyl cyclases, so they all have the same basic structure. And I'll stress once more that this is a picture that applies for the membrane-bound adenylyl cyclases, but not that soluble adenylyl cyclase. Okay, so once again, let these two lines represent the inner and outer leaflet of the phospholipid bilayer. Okay, adenylyl cyclase enzymes are made up of a single polypeptide. Okay, so the amino terminus of this polypeptide is in the cytoplasm. Then what you have is a special domain known as the transmembrane domain 1, which consists of 6 membrane-spanning alpha helices, which have all been put together. Okay, so they all sit as a cluster together. So this cluster of 6 membrane-spanning alpha helices, highlighted in pink here, this is known as the transmembrane domain 1. And for short, transmembrane domain 1 is often abbreviated to TMD1. So this stands for transmembrane, which is the TM. Okay, and then the D is for domain, so transmembrane domain 1. Then between, then you have an intracellular loop, which we'll come back to in a moment, and then another one of these transmembrane domains, so another cluster of 6 membrane-spanning alpha helices, like so, and we'll colour these in red again as well. So 1, 2, 3, 4, 5, 6. Okay, and this cluster of 6 membrane-spanning alpha helices is known as the transmembrane domain 2, or the TMD2. Okay, and then you have a cytoplasmic carboxylic acid tail. Okay, so here's the carboxylic acid terminus of the polypeptide. Now, the transmembrane domains, they just anchor the whole thing at the phospholipid bilayer. Okay, they are not the active enzyme portion. Instead, the active enzyme portion is going to come from this domain that we've got here, this loop between transmembrane domain 1 and transmembrane domain 2, and also from this carboxylic acid tail here. Okay, right, so let me give you some nomenclature. This loop between transmembrane domain 1 and transmembrane domain 2 is known as the C1 domain. Okay, and then the carboxylic acid terminal domain is known as the C2 domain. Okay, now, the C1 domain is further divided into 2 separate portions. Okay, so we have this first portion here, which I'm going to highlight in orange. Okay, and this is known as C1a. Okay, the C1a domain. And then after that, we have this portion that's in green here, and this is known as the C1b domain. Right, similarly for C2, we can divide C2 up into 2 portions. I will once again highlight C2a in orange, so this portion is C2a, and then we'll have C2b, I think, in blue. Okay, right. So, those are the important domains of our adenylyl cyclase enzymes, okay, and all of the 9 membrane-bound adenylyl cyclase enzymes have this same structure. Okay, so what we now want to talk about is how you activate adenylyl cyclase enzymes, and to do that, we need to discuss where the actual active enzyme portion is. So basically, in my drawing, we don't actually have the active portion yet, because the active portion is going to be made up by the dimerization of C1a, this orange portion, with C2a in orange as well. That's why I highlighted both of them in the same color. So in order to activate this enzyme, you need to bind C1a with C2a, and when they come together, that then will make the active adenylyl cyclase enzyme, which will catalyze the cyclase reaction. Okay, so let me now show you an activated adenylyl cyclase then. And I'm going to have to use a little bit of artistic license here now to actually show C1a dimerized with C2a. Okay, so here we have the first transmembrane domain, so I'll just colour that in again so that we're consistent here. So here are 6 membrane-spanning alpha helices clustered together. Okay, so this is called transmembrane domain 1. Then we have this C1 domain, which is this loop between transmembrane domain 1 and then transmembrane domain 2 over here. Okay, and I'll colour transmembrane domain 2 again in red here. So here are these 6 membrane-spanning alpha helices. So this is TMD2. Okay, right, and now I want to show C1A dimerized with C2A. So firstly, let's highlight up C1A. Okay, so once again, here is C1A in orange, and then we'll have C1B in green here. Okay, so this portion here is C1B. Right, and now what's going to have happened is C2A has to now be dimerized with C1A, and this is where I'm using the artistic license. I'm going to stretch C2A out somewhat. Okay, so here is C2A, also in orange here. Okay, now nicely dimerized with C1A, and then finally we'll have C2B here in blue, and then finishing off with the carboxylic acid tail down here. Right, so this now is the active adenylyl cyclase enzyme. This portion here is the active catalytic site, which will actually be performing the cyclase reaction, whereas this is the inactive one. So let's now just have a brief discussion of the cyclase reaction itself. What do adenylyl cyclase enzymes do once they have been activated like this? Well, basically they take in adenosine triphosphate, ATP, and they convert it to cyclic AMP and pyrophosphate. So let's draw a few little cartoons for this reaction. So we'll start with adenosine triphosphate. So to understand adenosine triphosphate, we need to firstly understand what adenosine is. Okay, so adenosine is the name for ribose, okay, which is a pentameric ring structure like so, okay, bound to an organic base, adenine. Okay, right. So when you have adenine, the organic base adenine, bound to a ribose sugar, which is being shown here in blue now, that is called adenosine. Okay, right. It's what is known as a nucleoside. So I'll just put this keyword here. Okay, so a nucleoside, not a nucleotide. A nucleoside means an organic base such as adenine bound to the ribose sugar. Okay, so when you just have a ribose sugar with an organic base, that's called a nucleoside. So adenosine is an example of a nucleoside. You could also make other nucleosides by having a different organic base here. Now, we want to convert the nucleoside into a nucleotide now. Okay, and a nucleotide is just a nucleoside with at least one phosphate group stuck off. Okay, so we'll stick one phosphate group there, and that turns us into a nucleotide. But you can add more than one phosphate group, and it's still called a nucleotide. And we're going to add 3 phosphate groups, adenosine triphosphate. Okay, so these 3 purple blobs here, these represent the 3 phosphate groups that we've stuck off the alcohol group that comes off the 5th carbon of the ribose sugar. Okay, right. So adenosine triphosphate is an example of a nucleotide. So a nucleotide, once again, is just a nucleoside with at least one phosphate group on it. But you can have more than one phosphate group, so you can go up to 2 and 3 phosphate groups, and that is still a nucleotide. Okay, so this is adenosine triphosphate, and for short, adenosine triphosphate is abbreviated to ATP. Okay, right. So adenosine triphosphate is the substrate for the active adenylyl cyclase enzyme. What are the 2 products? Well, the famous product is cyclic AMP, or abbreviated to CAMP, and this in full stands for cyclic adenosine monophosphate. Okay, so we are in a perfect position to understand what adenosine monophosphate is. We know what adenosine is, that's just adenine plus ribose. Okay, and I was about to write that little equation here. Ribose plus adenine makes you adenosine. Okay, to create adenosine monophosphate, all you do is stick a single phosphate group off the alcohol group that comes off the 5th carbon up here. Okay, how do we turn it into cyclic adenosine monophosphate? That's a little bit more of a question. So, firstly, let me just tell you about the nomenclature for these 3 phosphate groups within the adenosine triphosphate molecule. So the first phosphate group within the adenosine triphosphate molecule is called the alpha phosphate. The second phosphate group is called the beta phosphate, and the third one is called the gamma phosphate. So we have the alpha phosphate, the beta phosphate, and the gamma phosphate. Okay, in this cyclase reaction, as it's called, okay, so this is the reaction catalyzed by adenylyl cyclase enzymes. Okay, you take in adenosine triphosphate and you cut off the beta-gamma phosphates here. Okay, so we've got another beta-gamma here, damn it. But never mind, we're going to cut the bond between the alpha and the beta phosphate there, and what we'll get is two phosphate groups being released. Okay, so this is the beta phosphate still bound to the gamma phosphate, and this is our other product of the cyclase reaction. Okay, so this is called a pyrophosphate molecule. So when you have two phosphate groups bound together, it's called pyrophosphate. Okay, and for short, pyrophosphate is often abbreviated to PP for pyrophosphate, and then people usually put an I there. It's the same as when people abbreviate inorganic phosphate to PI. Okay, so that's the same spirit. Okay, right. Then we would have adenosine monophosphate to create cyclic adenosine monophosphate. What we're going to now do is we're going to bind our alpha phosphate to the alcohol group that comes off the third carbon of our ribose sugar. Okay, so let me show you doing this. So firstly, let's just draw out what we understand. Here's our ribose sugar. Here is our adenine organic base here, so that's just adenosine. So I'll colour these in again. Here is our organic base adenine in green. Here is our ribose sugar in blue. Okay, and then coming off the fifth carbon, we have this phosphate group still, the alpha phosphate, and we're going to bind the alpha phosphate to the alcohol group that comes off the third carbon down here. Okay, and that creates us a new cycle within the structure. It's still adenosine monophosphate because we have a single phosphate group coming off adenosine, but you know, a normal adenosine monophosphate would not have this bond here, it would just have a single phosphate group coming off up here. Okay, we've bound it to the third carbon's alcohol group and therefore created this extra cycle within the structure, and that's why this is called cyclic adenosine monophosphate. Okay, so adenylyl cyclase enzymes, once activated then, produce cyclic AMP. Okay, the next thing to discuss is how alpha S-GTP subunits are going to activate adenylyl cyclase enzymes. Okay, and then what we want to discuss is the different types of adenylyl cyclase enzymes. Okay, so I've told you that there are 9 membrane-bound adenylyl cyclase enzymes. They're grouped helpfully into 4 different families, and I want to just discuss those 4 different families. So firstly, let me tell you how the alpha S-GTP subunit binds to and activates the adenylyl cyclase enzymes. So basically, our alpha S-GTP subunit, okay, which I'll draw here, is going to come along, it's going to find the adenylyl cyclase enzymes, here it is, okay, with guanosine triphosphate bound to it, and what it does is it binds both to the C1 domain and also to the C2 domain. Okay, and what it does is it brings C1 and C2 close together, and this promotes the dimerization of C1a with C2a. So basically, it's going to come here, it's going to bind some portion of C1, so it's going to grab onto the C1 loop here. It's then going to bind onto some portion of C2, so it's going to grab the two, and it's going to pull them close together, and this promotes the dimerization of C1a with C2a to form the active adenylyl cyclase, and that's how the alpha S-GTP subunit is going to activate the adenylyl cyclase enzyme. Okay, right. Next up, I just want to talk briefly about the 4 different families of adenylyl cyclase enzymes, which will be helpful in giving you some understanding, a broader understanding of these 9 adenylyl cyclase enzymes, because at the moment that's just a fact with no meaning to it. Okay, if we categorize them into families based on what signaling pathways affect them, then it means something more. Okay, and the first thing I should say is that alpha S-GTP subunits activate all 9 adenylyl cyclase enzymes, which are membrane bound and therefore look like this. So it's got very broad specificity, basically. Okay, so now let's talk about the 4 families of adenylyl cyclase enzymes, and I'll try and squash this in up here, which will probably be a mistake. Okay, so firstly, the first family is the calcium calmodulin activated family. Okay, so this is the calcium calmodulin activated family. And there are 3 members within this family, okay? There is adenylyl cyclase 1, adenylyl cyclase 3, and adenylyl cyclase 8. Now, what does it mean to be in this family? It means that you are calcium calmodulin activated. So basically, if calcium goes up in the cytoplasm nearby one of these 3 adenylyl cyclase enzymes, then the calcium will bind to calmodulin, and calcium calmodulin complexes can activate these 3 adenylyl cyclase enzymes, but not the other 6, okay? Only these 3, and that will promote the dimerization of C1a with C2a. Okay, so those 3 are going to be activated by intracellular calcium. Next up, we have the G-beta gamma stimulated, okay? And this is a little bit more relevant for our pathway. We're not talking about calcium here, okay? So we're not interested in these 3, but this one's more interesting, okay, as far as our pathway is concerned. This family contains 3 members again: adenylyl cyclase 2, adenylyl cyclase 4, and adenylyl cyclase 7. Okay, so I have told you that the alpha S subunit with GTP bound is capable of activating all 9 adenylyl cyclase enzymes. Okay, however, these 3 are special because the adenylyl cyclase 2, adenylyl cyclase 4, and adenylyl cyclase 7, if they have been activated by an alpha S GTP subunit, then what can happen is the beta gamma complex can come and bind on top of them. Okay, so let me show this. Here is the beta gamma complex. Okay, so imagine this adenylyl cyclase enzyme here is now either one of these 3. Okay, we've had the activation by the alpha S GTP subunit, and that's very important. Beta gamma is not capable of activating it on itself. It has to have already had the alpha S subunit do its job. Okay, so you've got one of these 3 adenylyl cyclases. Okay, the alpha S subunit has bound and has activated the adenylyl cyclase. Now what's going to happen is the G-beta gamma complex is going to come and bind as well to the adenylyl cyclase, and this will increase the activation of the adenylyl cyclase even more than when it just had the alpha S GTP subunit bound to it. So in the case of these 3 adenylyl cyclases, and only in the case of these 3, the beta gamma complex can potentiate the which means make bigger the activation that the alpha S GTP subunit delivers to them. So to summarize, the alpha S GTP subunit activates all 9 adenylyl cyclase enzymes, but the activation that it produces can be enhanced in these 3 adenylyl cyclases, the G-beta gamma stimulated adenylyl cyclases, by the binding of the beta gamma complex to the adenylyl cyclase, which has already got the alpha S GTP subunit bound to it. The beta gamma complex is not capable of activating these on its own. It's only capable of enhancing the activation brought about by the alpha S GTP subunit. Okay, right, so I hope that's clear. Next up, the next family, which I don't think I'll try and squeeze in there, I'll go down here. Okay, so the next family is called the calcium and G-alpha I inhibited family. Okay, so this family contains 2 adenylyl cyclase enzymes, namely adenylyl cyclase 5 and adenylyl cyclase 6. Okay, so AC5 and AC6. Now, in contrast to the calcium calmodulin activated adenylyl cyclases, which are activated by calcium binding to calmodulin and forming calcium calmodulin complexes, which then interact with the adenylyl cyclase, these 2 are directly inhibited by calcium. Okay, so if calcium is high in the cytoplasm around either adenylyl cyclase 5 or adenylyl cyclase 6, the calcium on its own, not through calmodulin, on its own can inhibit the adenylyl cyclase 5 and the adenylyl cyclase 6 enzymes. Okay, in addition, these are the two adenylyl cyclases which are actually inhibited by the G-alpha I alpha subunits of heterotrimeric G proteins. In fact, it's broader than that. They're inhibited by G-alpha I1, G-alpha I2, G-alpha I3, all of the I's, but they're also inhibited by G-alpha O, which remember I told you had very similar properties, and G-alpha Z. Okay, so these are the five alpha subunits of heterotrimeric G proteins which can inhibit adenylyl cyclases, and unlike the alpha S GTP, which has its effect on all of the membrane-bound adenylyl cyclases, they only affect certain adenylyl cyclase enzymes, and adenylyl cyclase 5 and adenylyl cyclase 6 are inhibited by these five alpha subunits of heterotrimeric G proteins. Okay, right. The final family then of adenylyl cyclase enzymes, I need a break soon. Okay, so is the forskolin insensitive family of adenylyl cyclase enzymes. Now, we have seen 8 of the 9 members, so there is only 1 member left that can possibly be within this family of forskolin insensitive adenylyl cyclases, and this is the adenylyl cyclase 9 enzyme. Okay, so firstly let me tell you what forskolin is. Forskolin is a drug which is not used clinically at all. It has no clinical value whatsoever. It would be most likely very toxic. However, it's a drug that is fantastically experimentally important. So in experimental science, this is an incredibly important drug, and the reason is that it activates adenylyl cyclase enzymes. It binds to adenylyl cyclase enzymes, promotes the dimerization of C1a with C2a, and therefore starts up the cyclase reaction. So it's an experimental drug capable of activating adenylyl cyclase enzymes. Now, it works on all of the adenylyl cyclase enzymes which are membrane bound, i.e. AC1 through AC8, AC1 through AC9, apart from AC9. So it doesn't work on adenylyl cyclase 9, but it works on the other 8, basically. Okay, so that's why adenylyl cyclase 9 is grouped into this separate family on its own, which is the forskolin insensitive family of adenylyl cyclase enzymes. Okay, right. So, we've seen now that the alpha S GTP subunit is capable of activating all 9 adenylyl cyclase enzymes, and in addition, the beta-gamma complex will enhance the activation of those 3 adenylyl cyclase enzymes which are in the G-beta-gamma stimulated family of adenylyl cyclase enzymes, which are the adenylyl cyclase 2, the adenylyl cyclase 4, and the adenylyl cyclase 7. Okay, this has resulted in the production of loads of cyclic adenosine monophosphate, CAMP. And this now is going to activate the next component of this signaling cascade, which is protein kinase A. Okay, so in the next video what we'll do is discuss protein kinase A.